All right, last video for this week. We are going to talk about scientific method and some experimental design information. So, scientific method is a process for discovering answers to questions based on observations. So, scientific method is pretty complex as you get into more and more details, but the kind of basic process is observation, question, hypothesis, experiment, collect, analyze the data, and then make a conclusion, and then compare that to your hypothesis. And if it's inconsistent, you got to revise that, you got to make a new hypothesis. If it's consistent and supports it, then you need to reproduce the experiment several times. And ideally get other people to reproduce the experiment too. And if that hypothesis is supported over and over and over and over and over again for a long enough period of time without getting any evidence conflicting it, it can be accepted as a scientific theory. This is not the same way we use theory in our kind of common everyday language, though. So the basic steps: observation. This is something that we see in nature, something you experience, thoughts that you have based on things that you've read. So you make an observation. I see this happening. And then you form a question. Why is this happening? Next step is a hypothesis. A hypothesis is an informed, educated, logical guess. I also like to think of a hypothesis as a possible answer to the question. You are making this hypothesis as a possible answer or explanation for that question. If this is true, it's because of whatever. So then you need an experiment to test that possible answer or explanation. You need to test that hypothesis and see if it is, in fact, a good answer to that question. So you design the experiment, you conduct the experiment, and then you collect and analyze the data. You've got to collect it, and then you analyze it critically. This is the both sometimes boring and really exciting. Kind of depends on where the data takes you. But it does need some very, very critical analyzation, and this requires a lot of statistical analysis, which can be really complicated. Based on that analysis of the data, we are going to form a conclusion. Does that evidence, did the experiment and the evidence we got from it support our hypothesis, support that answer, or does it conclude that that is not an accurate answer to the question? If that evidence, if our conclusion rejects the hypothesis, it says, no, this is not an accurate answer to the question, then you form a new hypothesis. You come up with a new possible answer to that question. And then you form a new experiment. And if it does support the hypothesis, says, yeah, this seems like it could be a very accurate answer, you repeat the experiment. You want to replicate it. Because sometimes you get weird one-off conclusions and experimental results that are not really consistent, so you replicate it. You do it again, and again, and again, and again. And then you keep doing it. And then you get other people to start replicating it, using your exact same experimental process. And that's where we start getting scientific consensus. If enough people doing this experiment frequently enough are getting consistent results every single time, then that is more likely to be an accurate hypothesis, an accurate explanation. It's kind of like if you're shopping online and you're looking at reviews. Some items, whatever you're looking for, maybe there is a perfect 5-star rating that is based on 30 reviews, and you're comparing it to an item that maybe only has like a 3.9 star rating, but it's based on 50,000 reviews. Now, you're not necessarily always going to choose the item with more reviews. Sometimes you take a chance on the other one, but generally speaking, people are going to opt for an item that has more reviews, even if it's a lower star rating, because that's more data explaining how good of a quality that product is. So once we've replicated that experiment enough, a model comes into play. So when that data supports the hypothesis multiple times, the hypothesis can become a working model. This is now accepted in the scientific community as a very, very accurate explanation of what's going on, but this still needs to be tested over and over again, pretty much forever. And if that goes on long enough, we get scientific theories or scientific laws. So the law of gravity is pretty well known at this point in time. So scientific theories and scientific laws. Scientific theories are not the same type of theory that we use in our common everyday language. The way we use theory in our everyday language is actually more similar to a hypothesis, that possible explanation. So I want to be very clear about that. All right, so there's a lot that goes into designing experiments, and one of the big things is variables. These are things that could change and can be measured. Generally speaking, we need to design experiments to look at one variable at a time that seems to be significant in that explanation to answer that question. So we have both independent and dependent variables. Independent variables, this is what's controlled by the experimenter, the researcher, the scientist. It is the variable that we are intentionally changing or removing from the equation to see if it really is having the effect that we think it is. The dependent variable is what we are measuring in response to whatever we did to the independent variable. This is the data we are collecting. So the dependent variable depends upon what happens with the independent variable. So if somebody's conducting an experiment to see if increased levels of saturated fatty acids would cause increased levels of serum cholesterol, the intake of those saturated fatty acids would be the independent variable. That is what's being controlled or removed from their diet. Either it's removed or it's given in varying levels, and the serum cholesterol would be the dependent variable. That's what we are measuring to see how it changes in relation to whatever we did to the independent variable. And then we have experimental control groups. This is a duplicate of whatever group we're experimenting on, where we did change the independent variable, that has to be the exact same as that group in every way except for the independent variable. So the purpose of the control is to ensure that any changes that are observed are due to that independent variable. They are due to that one thing we changed and not something else. And there are also controlled variables. So this is where, if you're measuring, again, that dietary level of saturated fatty acids, how it affects serum cholesterol, you want to make sure that these people are otherwise not changing their diet in any way. All right? You want to make sure that every other aspect of their diet is the same. Because if these people say we are having a group that is eating increased levels of saturated fatty acids in their diet, and maybe another group that is having lower levels of saturated fatty acids in their diet, you don't want them to also increase or decrease their sugar intake at the same time, or increase or decrease their fiber intake at the same time, because those are all things that could potentially impact serum cholesterol levels. So you don't want those things to change, because then you don't really know, was it the saturated fatty acids that changed serum cholesterol levels? Or was it the fiber or the sugar levels or anything else like that? So you want everything else in the experiment and the groups to remain exactly the same. You just want to change one thing at a time. So our control group in this situation would be individuals of a similar age to what we're experimenting on, similar age, gender, and weight that don't have that increase in dietary saturated fatty acids. Whereas our experimental group is the group that we are increasing their dietary saturated fatty acids. But again, neither group should be changing their diet in any other way for the duration of the experiment. All right, so we're going to look at a slightly different aspect of studies now. So variability in different types of scientific studies. Because not every study, not every experiment is created exactly the same, and there are a lot of reasons why we would need to create different types of experiments. So we have crossover studies, blind studies, double-blind studies, and then double-blind crossover studies. So variability has to do with how spread out or closely clustered a set of data is. So basically, how similar is all of the data, or are we getting wildly different results from different sources? So human populations, we have so much variation in our genetics and environment. And so that can make it really, really, really difficult to accurately determine how specific single things change different aspects of our lives. So in order to show an actual significant, a real difference between experimental and control groups, we need to include a really, 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 really large number of very similar subjects, because then you are reducing the impact that the genetic and environmental variation has on whatever you're measuring. All right, crossover studies. This is where each subject actually participates in both the experimental and control groups. So then we are measuring the individual's response to the treatment versus themselves. This is really effective when there's a lot of variability within a population. And this is also going to involve something called a washout period. This is when the subject is not getting any treatment under the study, and any effects of a previous treatment should be eliminated. So basically, it's a period of time where they are just kind of letting whatever treatment it is decrease in effectiveness. So there's advantages and disadvantages to doing this type of study. So comparing an individual's responses to their own control values can be really, really beneficial for certain things because of varying genetics or environmental factors. But it can also skew some of the data depending on exactly what we're trying to measure. So this is a very controlled process. So say they start with the treatment, the experimental group first, and they receive the treatment for a certain amount of time, and then they switch to the other group and receive whatever the other treatment is based on that independent variable. So this is generally most relevant when the outcome is reversible with time. So things like symptoms. So symptoms as a result of certain medications. So this could be side effects, or maybe their symptoms are trying to be treated by that medication. So control group is the first half of the experiment, and then they become the experimental group in the second half, and then they can flip-flop. So some advantages to this are that all of the subjects serve as controls, and the possibility of errors is reduced a bit. And this can mean that we need a smaller sample size in some cases. all subjects receive treatment at least part of the time. So this comes into consideration with ethics boards, is there are some conditions where it is considered to be unethical to withhold treatment from a group of people. So I'll kind of give an example of that here in a moment. And then statistical tests, assuming randomization can be used. So kind of randomizing who's getting what treatment when can help with statistics. And we can also keep this blinded. And we're going to talk about blind studies here on the next slide. Some disadvantages to crossover studies are that all subjects are going to receive that placebo treatment or alternative treatment at some point. Again, this can create some ethical concerns if it is a serious condition that we are evaluating. So, for example, there are a lot of medications, vaccines even are tested this way, where if we have a treatment, either a vaccine or some sort of medication for a particular disease or condition that we already have, it exists, and we know it works. If you are testing something new, a new type of vaccine for that pathogen, a new drug to treat that condition, it is generally considered unethical to have a control group that does not get the treatment or vaccine at all, because we know we have something that works. So generally in those cases, we actually compare new vaccines or new medications to the existing ones that we know work. So we are not putting anybody at additional risk by having them in a group that is not receiving any treatment whatsoever. So the receiving placebo or anything like that can be an ethical concern. So depending on what type of study is being done, crossover studies are not always the best case scenario. They're not always the best type of study to use. The washout period, another disadvantage for this type of study, is that the washout period can be a really long time, depending on how long it takes for, that drug to leave their system or anything, or it can be completely unknown. We don't always know what the washout period is going to be. So sometimes they just have to take an educated guess at what point the study ends. And then this type of study also cannot be used for treatments that have permanent effects. So surgical procedures and stuff like that would fall under that, because those could have very permanent effects. All right, blinding studies. So we can have single blind studies where just the subjects, the people participating in the study, do not know if they are in the treatment or placebo group. This is good because it reduces the placebo effect, which we will talk about more in a little bit, but basically it's proof that our mind is a very powerful thing. Thinking that you're going to get better with a certain treatment or not get better can actually influence how you feel as a result. So a blind study where the subjects do not know what they're getting kind of reduces that unconscious bias. But there can be problems because the researchers know who's getting what. And so the researchers can subconsciously treat each subject different because they know what they're getting. This can be prevented by having a double-blind study. So this is where neither the subjects or the researchers know who's getting what treatment, either the treatment or the placebo. A third party who is not involved in the experiment outside of just data analysis is the only one who knows which group got the experimental treatment and which one got the placebo. But that third party that's doing the data analysis is not involved in any other aspect of the experiment. So they are not giving the treatments, so they are not able to potentially treat the subjects differently or anything like that. Generally speaking, double-blind studies are considered the gold standard, but they're not always possible. Even blind studies are not always possible. Sometimes you have a pretty rare condition or disease, and it's unethical to give somebody a placebo if it's a super, super severe condition. So just off the top of my head, for example, prion diseases. These are very odd, basically contagious infectious proteins. They are considered to be lethal, non-curable diseases. They're really serious, but they're also very rare. So you're not going to get a big enough sample size to do a lot of research. And also, if you were to get, say, 5 people that have a prion disease and you're testing a new medication that might help either slow the progression of the disease or potentially cure it, it is not ethical to withhold that treatment from anybody, right? Even though we don't know if it works, the potential that it could work means that it is unethical to put anybody in the placebo group. And so then you would not have a blind study at all because everybody's getting the treatment. Everybody knows that everybody's getting the treatment. So there are always exceptions to this. But whenever possible, we try to do blind or double-blind studies. Then we can combine those into a double-blind crossover study or even just a blind crossover study. So same idea, control group in the first half becomes the experimental group in the second half and vice versa, but nobody knows who's getting what at what point in time except for that third party. So we can combine those to get an even better result from our studies that ideally eliminates as much of that subconscious or even conscious bias as we possibly can. All right, placebo versus nocebo. So placebo is Latin for I shall please. So a placebo drug or treatment or anything, this is an inactive or medically inert treatment or substance like sugar pill or a starch tablet. Does not have any active therapeutic properties. It has no medical effects. It is a fake treatment essentially. And it is very commonly used in research and clinical trials to compare the effects of real treatments against the effects of a perceived treatment. The placebo effect is when a patient taking the placebo thinks they are taking the actual medicine, and because of the power of our mind, there is a positive change in their health because they think they are taking the actual medication. So, again, these blind and double-blind studies remove that placebo effect because then patients don't know if they are getting the placebo or the actual medication that's being tested. So that kind of helps remove this power of the mind thinking that you are going to get better or not get better. So it's a really interesting effect, and there are some rare cases I've heard of where the placebo effect is actually used as a treatment. These are very rare cases, but it's interesting that it is sometimes used as a treatment to basically get the mind to trick itself into getting the body better. Then the nocebo effect, Latin for I will harm. This is an inert substance or therapy that should be ineffective, but causes symptoms of ill health. So this is when you warn people a drug they are taking may have specific adverse side effects. Those people frequently will report a higher incidence of the side effects than a similar group of people who were not warned. So again, this is that power of suggestion from our mind, telling people, hey, you might have nausea, vomiting, insomnia, a rash, anything like that. They are more likely to report that they are nauseous or vomiting frequently than people who were not told of those possible side effects. Now, obviously, there are some ethical concerns there. People should be aware of the potential problems of medications they are taking. So that can be kind of tricky. But I think the important thing to remember with these adverse side effects is everybody reacts to medications differently. Just because the side effects are possible does not mean that you are necessarily going to get them. So this can take, again, a little bit of kind of working with your brain to convince it that you are not guaranteed to get those side effects. So again, this is an ill effect caused by the suggestion or belief that something is going to be harmful. So it's kind of the evil twin of the placebo effect. kind of the same idea. It's all based on the perception of the patient. But again, obviously, we have some ethical concerns here with this nocebo. Withholding information about adverse side effects can be dangerous, and so it is generally frowned upon to do that. And again, with these placebo treatments, as I mentioned earlier, in some cases, if we have a treatment for something, for a disease or condition or vaccination against a certain pathogen that we know works, we have ample evidence over decades to centuries that it is going to be effective, it is considered in some cases to be unethical to withhold any type of treatment and to have anybody in a placebo group when we are testing a new medication or vaccine. All right, so a lot of these scientific designs, we have to take those ethics into consideration also, which has not always been the case in science historically, which is really horrific in some cases. And so I am happy with the progress that we've made over the years to make science a lot more ethical. But that wraps things up for this week. If you have questions, definitely let me know, and I will see you all in lab on Friday.